Hey guys, so today we're going to be going over chapter 5. We're going to go more into detail as to what categorizes a bacteria cell and a eukaryotic cell. So let's get started. So we've talked about what two classifications of cells there are. There's prokaryotic and eukaryotic, and in this chapter we're going to go more into detail about it. So let's start off with prokaryotic. So prokaryotic are bacteria and archaea. So the archaea are like the bacteriums that live in the extreme environments, such as extreme cold or extreme heat, um, very acidic or very salty environments, just the extremities of our environment. They lack a DNA nuclear membrane, so their DNA is not in one specific area, it's throughout the entire cell. They're also relatively small, especially compared to eukaryotic cells. They are smaller. And they also have a very complex cellular envelope. So it's made up of multiple things, such as a cell membrane, a cell wall, and sometimes additional layers. And we'll go more into detail about what those outer layers are. And then also, they have a very small genome. It's very compact and it's very simple compared to eukaryotic genomes. And then also they have very tightly coordinated cell parts. So everything within the cell is very compact. I don't know how else to say it. And then also everything has a purpose. And the reason why is because they have to reproduce quickly. So they need to be a very tip top shape in order for them to be successful. And then with eukaryotic cells, these are the all other cell types that aren't considered prokaryotic. So this is such as like our cells, plant cells, and then also other species such as like chimps, monkey, dogs. They're all eukaryotic. And then the biggest difference from eukaryotic and prokaryotic is how they store their DNA. So eukaryotic cells, we have our DNA within inside a nucleus, whereas bacteria don't. So a ideal bacterial cell will have the following. So they'll have a cellular membrane, they'll have a cell wall that is made up of peptidoglycan, and then they'll have various outer membranes, primarily made up of lipopolysaccharides. And then sometimes they'll even have a capsule, they'll have a flagellum, and then a nucleoid. And we'll go into these a little bit more throughout the lecture. So, what defines the existence of a cell? So, the membrane is a defined structure of the cell. Like it is the outside of it. It's what keeps everything in its place, and then also it separates the cytoplasm from the extracellular from external medium. So from the outside. So it's like the wall between cell and environment. And then also the cell membrane plays a huge role in mediating transportation between stuff on the outside and the inside of the cell. So it's kind of like border patrol for the cell. Also, they have a lot of proteins. So these proteins, they provide structural support. They also detect the environment as to like, is the environment friendly or not? And then also it secretes virulence factors and communication signals, and then it allows transport between outside and inside of the cell through the cellular membrane. And then also it has to do with energy storage and transfer. So with the cell membranes, how does the bacteria actually use it to its advantage to protect it? So the cell wall makes the cell very rigid, and with making it more rigid, it can withstand intracellular turgor pressure that builds up as a result of osmotic shock. So it it creates this balance, and with the fact that the cell wall is very rigid, the cell doesn't collapse in on itself because of pressure and the osmotic shock. So the osmotic shock has to do with water movement, and that's pretty much all I want you to know. But just know that turgor pressure plays a huge role in allowing the cell not to collapse in on itself. And then, do eukaryotic cells have cell walls? In general, no, but there is, of course, exceptions. So eukaryotic cells that do have cell wall is some, I think almost all fungal species have cell walls, as well as plant cells also have cell walls, but eukaryotic cells that don't have cell walls are such as like ours. Like, we do not have cell wall, we just have a cellular membrane. So now we're going to go more into detail as to what makes these outer layers of a bacteria. So the biggest thing is peptidoglycan. Peptidoglycan itself is actually very specific to bacteria only. You don't find peptidoglycan in any other cellular structure. You only find it in bacterial structures, and it's very unique. So what it is it creates these parallel polymers of sugars, essentially called glycans. And these glycans, they form chains with proteins, so with peptides, excuse me. And then there's these repeating units of NAG and NAM. And with that, the strands are linked with peptide bridges. And then that is what actually forms the bacterial cell wall. So it's just this big link of sugars and amino acids, essentially. So the biggest thing I want you to get from this slide is the fact that peptidoglycan is unique to bacteria, and also there is a lot of linkage that happens between multiple units. And then also, I don't want you to know how peptidoglycan is synthesized, that's more biochemistry, but just know that there's a lot of genes that go into having to make peptidoglycan. And then also, with peptidoglycan being very specific to bacteria, it is a huge target for antibiotics. So whenever you take antibiotics, you want to make sure that your medicine is actually only hurting what is making you sick. You don't want it to hurt your patient. So the fact that humans, we don't have peptidoglycan, we know for a fact that antibiotics will only attack peptidoglycan. So the antibiotics won't actually be hurting us. And that is a big thing for like penicillin, for vancomycin. Those specific antibiotics actually target the peptidoglycan within bacteria. And then the way that happens is specifically with vancomycin, what it does is that it prevents the formation of it by preventing bonds happening. So it interrupts the synthesis of peptidoglycan of the bacteria, which is really nice, because with the fact that the peptidoglycan isn't being made for the cell wall, the bacteria is no longer protected by its cell wall. And then also, vancomycin is considered like a last resort drug because you don't want to build resistance. So a lot of times if somebody has like MRSA, they will give them vancomycin because MRSA is, specifically that staph infection, is resistant to methicillin. So we'll give vancomycin instead. Just a little side note. So, like I said earlier, like the widespread use of antibiotics can evolve resistance within a strand. So there's specifically resistance in the cases below. So beta-lactamase specifically cleaves penicillin. So the beta-lactamase is made by the bacteria and it cleaves the penicillin. So with the fact that penicillin is no longer penicillin because it got broken up, it actually prevents penicillin from doing its job. And then cells that are specifically resistant to vancomycin, what they do is that they have this alternate enzymes that actually add something to it. And then with the fact that it is altered, the bacteria is no longer affected by the presence of vancomycin. So now we're going to transition into what specifically makes specific bacteria cell walls and outer membranes more specific. So let's talk about gram-positive bacteria. So we kind of dabbled in this a little bit, but we're going to go more into detail. So gram-positive bacteria, this is generally how they look in the diagram. So they have a very thick cell wall and they have multiple layers of peptidoglycan, as you can see right here. So these blue purpley squares, this is the peptidoglycan within its cell wall. As you can see, it's very, very thick. And then also it has just a little bit of glucochains over here as compared to gram-negative, which only have a single layer of peptidoglycan. So they have a very thin cell wall, but they have an outer membrane as compared to gram-positives. And then with the fact that they have an additional outer membrane that has a lot of lipopolysaccharides, it helps it in its virulence. So with the fact that it has this thicker outer membrane as compared to gram-positives, it helps it be resistant to phagocytosis by our immune cells. Okay, so now we're going to talk about, oh, it's not going. There we go. So now we're going to talk about bacteria that don't really fit into this this gram-positive or gram-negative structure, and they're called mycobacterium. So they are considered gram-positive, but as you can see, their cell wall and outer layers are completely different compared to gram-positives and gram-negatives. So the biggest thing with these guys is that they have this myocholic acid. That's the biggest thing that will distinguish them from gram-positives and gram-negatives. They still have a cellular membrane and they do have peptidoglycan, but the big thing is this myocholic acid. So this is specific to this cell type. So definitely know that, guys. So if I were to give you these on a test, so if I were to show you this figure and then this other figure and then the last figure, I would want you to be able to distinguish between them, as well as tell me something that is, tell me some descriptive factors of each of their outer membranes. So such as, oops, wrong way. Such as like gram-positives, they have a thick cell wall that have multi-layers of peptidoglycan. And then as well as like an example of that is like strep pyogenes. And then gram-positive, it has a very thin cell wall with a single layer of peptidoglycan, but in addition it has an outer membrane made of lipopolysaccharides. And the fact that they have these polysaccharides, it helps with resistance to phagocytosis. And then also with the last one, state that they are generally considered gram-positive, but they have an envelope, their cellular envelope, that is extremely thick and complex. So if you see like this entire thing right here is their envelope. Majority of this figure is their envelope, and know that within their envelope they have this acid, and an example of that is like tuberculosis. And with the fact that it has a very thick outer layer, is that it also helps uptake. It minimizes uptake of nutrients. So with these guys, the fact that they have such a huge channel for nutrients to get through, they grow very, very slowly. So also know something, just some little fun facts about them, okay? Okay, so let's move on. So, how does the bacterium actually protect their genome if they don't have a nucleus like us? So they have their cellular envelope, and that functions to contain and protect their genome. So, and then also bacteria, they have this filamentous nucleoids region that extends throughout the entire cytoplasm. So it's kind of like a little bungee cord system for their genome. So it's not just floating around willy-nilly. There are filamentous nucleoids throughout the entire cell internally that do help somewhat keep the genome grounded. So that is how they quote unquote protect and contain their genome. So with the nucleoids throughout the entire cell, it helps the bacteria organize their DNA. So that is how they organize their DNA, whereas in eukaryotic cells, we organize it within our nucleus. So it contains loops of DNA, and it's held together by DNA binding proteins. So as you see in the bottom figure, there's like these loops all the way around. So that's like the DNA. And then you see like those little dots on there. Those are the binding proteins. And then all these loops, they all connect, and then there's this place that's called the origin of replication, which is exactly where it's attached to the cellular envelope, and it's kind of like the cell's equator. So it's like the home. So there is some point of this DNA that will be bound to the cellular envelope of the bacterium, and that is called the origin of replication. So in this figure, it's right here at the top. But it won't always occur at the top. It can occur anywhere along the cellular envelope. But it will only occur in one place. And then also in bacteria cells, they have something called supercoiling. So this is how they keep so much DNA in such a small area, is that they use the function or the process called supercoiling. Whereas like in eukaryotic cells, we use something that are called histones. But for our class, I just want you to know that the nucleoid is a way of the bacteria organizing its DNA. It's held together by DNA binding proteins. And then also that all the loops will connect to one center point, and that one center point is called the origin of replication. And then the way that they keep their DNA so compact is through supercoiling. So that's the biggest thing I want you to get from this slide, okay, guys? Okay, so now let's go into bacterial division. So bacterial division, what will happen is that replication will begin at that replication origin. And then replication will proceed in both directions. So it will go from the right to the left. And then as it grows and grows and grows, it will reach a point of where it's done replicating. And then the cell will expand, so it will get bigger. And then separation will occur. So then they will divide into two separate cells. So once the DNA has been replicated completely into, now we have two complete genomes, the cell will divide and form two daughter cells. So the replication of the DNA termination site will trigger the growth of the dividing cell. And then at that point, that's called the septum. And then the septum will grow inward on either side of the cell, and then it will constrict it and seal off, and then you have something that's called separation. So that septation will be kind of like cytokinesis in eukaryotic cells. Okay, so spatial orientation of the septation will determine the arrangement of the cell. So cells form chains when they separate in parallel planes, such as Streptococcus. So whenever we actually in lab, we will see that strep actually forms chains whenever they are completely divided. So the cells will look like they are in a chain formation rather than looking like little individual cells. And then also they can even form clusters whenever the septate is in random orientation, which is how strep looks. So strep will look like, to me, they kind of look like little grapes. So we'll definitely see that in lab, and I'll point it out more. But just know that the way the cell is oriented will determine the end arrangement of the cells. Okay, so with that, I'm going to show you some different morphologies, meaning different cell shape and arrangement. So I definitely, from this slide, I definitely want you to know what cocci and bacilli and budding appendages mean. So cocci means circle, bacilli is kind of elongated, and then budding is like the elongation of a cell. You can kind of think of it as like a noodle if you want, or like a tentacle. So with cocci, there are multiple different types. So the basic shape is going to be cocci or bacilli. And then arrangement is going to be the prefix of the word. So cocci, if you just see coccus, that means that it's singular. If you see diplococci, that means that there's generally two cells side by side. And then if you have streptococci, it's in a chain. Or if you have staphylococci, so it's like in a cluster. And then tetrad is 4. And then kind of the same thing that goes with bacilli. So not so much cocci bacillus. You'll see more straight bacillus formations, so kind of like a little tube. And then diplobacilli is 2 of them together. And then streptobacilli is a chain of them. So definitely, big picture, just know the different shapes of cocci and bacilli and hyphae. Okay? Okay, so now let's go into the actual specialized structure of a bacteria. So bacteria do have special structures, and these structures do have specific tasks. So there are 3 main structures that I want to talk about. There's pili, and there's freebie, and then there's stalk. So the pili, this is what is actually used to attach to different substrates. So either to attach to different surfaces or to adhere to anything, essentially. So it helps with attachment. And then sometimes these pili can actually be used to transfer DNA between cells. And whenever there is a transfer of DNA from one bacteria cell to another, that's called a sex pili. And that's actually shown in this figure. So we have the donor cell, which is where the DNA originated from. He formed a pili with this receiving cell. So this is a sex pili. So DNA, once he replicated the DNA over here, he's going to transfer it to this recipient cell that didn't originally have it. So it's kind of like a file transfer. So if you're going to transfer like file from computer to flash drive, it's kind of like the same thing. But remember, bacteria do not have sex, okay? So just because it's called a sex pili does not mean that they had sex. They do not undergo sexual reproduction, okay, guys? They undergo something called conjugation, and we'll go more into detail about that later on in term. But just know bacteria do not have sex. Okay, so the next structure is a shortened version of the pili, and it's also used to attach to substrates. So they have the same function, but one is just shorter. And then lastly is the stalk. This is the membrane-bound extension of the cytoplasm, and this specifically will secrete adhesion factors, and they're called holdfast. Okay, so that's from this slide. That's pretty much all I want you to get from it, is exactly what's on this slide. So let's go to the next one. So let's talk about specifically flagellum. So there are multiple different types of flagellums, as you can see right here. So some of them have only one, some of them will have four, some of them will have them on either side, or all over the place, or none at all. There are multiple different types of flagellum, and for the most part, they all have to do with motility and chemotaxis. So I'm gonna actually fix the spaces on this because it's starting to irritate me. Okay, sorry guys. Let me just fix that. It's really bothering me. Okay, so rotary flagellum is specifically for motility and chemotaxis of a bacterium. So these are like little propellers that will drive the cell forward. So it's the motor for them, and the bacteria cell is the boat. Different bacteria will have a different type of flagellum, like I said right here. So there are 5 main types of different flagellum, and then also the type of flagella that they have can also enhance their virulence, such as with the Proteus species. They use the flagellum to actually swim up their urethra to infect the bladder, and then even continue to ascend into the kidneys. So a lot of times flagella is considered a virulence factor. Okay. So now we're going to go into a little bit more of their different structures. So there are thylakoids that conduct photosynthesis. So these are specified within photosynthetic bacteria, and they capture light. And with capturing light, they're actually able to make their own food and everything, so they don't have to rely on external sources. Also, some of them, they produce gas vesicles. So these are primarily in the aquatic phototrophic. So they use these gas vesicles for buoyancy, so they can also make sure that they don't go too deep or too high in the water, so as to keep them perfectly at the correct level to stay afloat, essentially. And then also, they have storage granules, so which is used to store energy. So kind of like how what we do, so like we store our energy as fat, they will store their energy such as like glucan or PHA. I'm not even going to attempt what PHA, like how to pronounce it, but it's essentially a biodegradable plastic. And then also there's some sulfur granules, so these are deposits of solid sulfur within the cytoplasm. And then the last one is metatomes, and these are microscopic membrane enclosed crystals of magnetites. So bacteria who specifically use this, they use it to aid in motility or to swim downwards towards a north magnetic pole. So they kind of use like the magnetic force in order to guide their motility and their direction. So it's kind of cool. Okay, so now we're going to transition into a eukaryotic cell. We're going to take a tour of a eukaryotic cell and go into specifically what each of these function, like what do each of these organelles do. Okay, so there is the endomembrane system, which is the ones that are inside the cell. So there's the rough ER. This is a continuation from the nuclear membrane, as we saw in the previous photo. So we have the nucleus, and then we have the rough ER. So as you see, they're very close together. So it's a continuation from the nucleus. With the rough ER, it does have ribosomes on it. So with the fact that it has ribosomes, it actually is a place of protein synthesis. So it makes proteins. Whereas the smooth ER does not have ribosomes on it. And it's specifically he's the director. So he sequesters substances away from the cytoplasm. So he's the one who tells substances to be like, you go here, you go there. He's the director. And then lysosomes, these guys contain enzymes that are specific to killing bacterium, and then also they can process organic materials for nutrition for the cell. And then the Golgi apparatus, this is a target vesicle, which for various destinations of the cell. So he also helps with um, telling substances what their final destination is. So kind of similar to the smoothie R. Now the nucleus is specifically for organizing our DNA in eukaryotic cells, whereas in prokaryotic cells it was the nucleoid. So the nucleus is specific to eukaryotic cells. So it's another organelle, and it contains our DNA, as well as sometimes it does contain some proteins. And then the nuclear envelope, which is what coats the nucleus, it has two main membranes. It has an outer membrane, which is the one that is connected to the ER, and then the space in between is continuous with the lumen of the ER. So just know that it's a nuclear membrane, and that it is also kind of like the gatekeeper as to what goes in and out. Then we have the nucleolus, which is the one that is specific to assemble the ribosomes. So like the ribosomes that are free floating in the cell, as well as the ribosomes that are connected to the rough ER. The mitochondria and chloroplasts. So in animal cells, we have mitochondria. We don't have chloroplasts, but chloroplasts is very important for photosynthetic cells, such as plants. So both of them they both yield energy. They are both considered like the powerhouse of the cell, and they evolved very early in history. And we actually kind of talked about this a little bit in the terms of the endosymbiosis theory. So that actually has to do with the second bullet point. So it was theory of that one cell, that the mitochondria or the chloroplast was uptook by a larger cell, but it wasn't digested or anything, and it started utilizing it, and then it became a mutualistic relationship. And then with the mitochondria specifically, it does have two membranes. It has an inner and an outer. So the inner membrane, sorry guys, it was bothering me. So the inner membrane specifically, it has a lot of folds, and those folds are called cristae. And then with that, it increases surface area. And then also with that, there are two internal compartments. There's the space in between the cristae, and then also the matrix. And then this is specifically the location of where ATP is synthesized. Chloroplast, so this has to do with plant cells, even some fungal cells. This is where they convert light energy from the sun into ATP during a light dependent reaction. And then with that, there are some thylakoid membranes that are located with the chlorophyll, and that is where specifically light is captured. So the thylakoid will capture the light energy, and then ATP and NADPH are used, and they reduce CO2 into sugar. And the region inside the inner membrane of the chloroplast is the stroma. And then this is where the actual reaction of where ATP and NADPH is made. So that takes place within the stroma. So now let's take a picture. Let's look at the picture of it. So I talked about the endosymbiosis. So theoretically, what we think happened is that with a mitochondria or a chloroplast, they were in the small cell. And then a larger cell came along. It's just like, oh, I'm going to eat you. But then what happened is that they ended up actually just utilizing those organelles for themselves. So that is the theory of how we got mitochondria in our cell, and how plants got mitochondria and chloroplasts in their cells. So lastly, with eukaryotic cells, these are specialized structures with eukaryotic cells. So some eukaryotic cells do have flagellum and then cilia. So these are bundles of microtubules, so like little fingers. They're very, very tiny extensions of the cellular membrane, and they have like this whip-like fashion where they're just constantly moving and wiggling. And then there's usually only like one or two flagellum, but with cilia there are just tens of thousands of millions. It's just endless. And then we have this next structure, which is specific to protists. So they have this thick, flexible pellicle that consists of membranous layers, and it's reinforced by protein tubules. So it's like their version of a tail. And then we have contractile vacuoles. These specifically will remove excess water from our cell by collecting fluids and contracting to expel them through a pore. So it's contractions, and then with those contractions, water or excess fluid is pumped out through the pores of a cell. Okay, guys, so that concludes chapter 5. So we went over specific parts of a bacteria cell, specific parts of a eukaryotic cell, and what specifically distinguishes them. We also went over the differences between a gram-positive and gram-negative and microbacterium with their cell wall and their outer layer, what makes them specialized. And then also we went over the basics of a bacterial cell division, and then also specialized structures of a bacterium and a eukaryotic cell. So I hope you all enjoyed this lecture. Have a good day.